right, so here's the overall results from the exam on Tuesday. Mean was a 78, like I announced, so a little bit higher than the other ones, no curve this time. 25% of you that took the exam got A's. So it was a good exam for a lot of you, not everybody, but a lot of you did significantly better on this exam. So that's good news. Still have a few people that need to take it. So, but do come by my office. I think tomorrow, I know, tomorrow I will have Zoom office hours, but I will be in my office here. So your choice, if you want to Zoom in, that's fine. If you want to come and look at your exam, I'm planning on being in my office in Henry probably from about 8:30 to 12:30. So all morning. I could probably stay a little bit longer than that if necessary. So stop by, send me an email if you want to schedule a time to come in and look that over. Okay. Let's see, anything else I wanted to talk to you about that? Okay. All right, so moving on. The next few weeks are going to be a little bit more rigorous, okay? We've got a big push here to Thanksgiving. And of course, after Thanksgiving, we're gonna be all online. So plan on, I know it's that time of the semester where you've got some fatigue, right? There's a lot of stressful things going on, the election, COVID, everything. Try for a couple more weeks here to stay focused and hopefully it will pay off. Okay, I emphasized last time as we move into this immunology material, pay a little bit more attention to the book. These concepts, especially as we move into adaptive immunity, it's going to benefit you to see them. Probably maybe the first time you see it, it's, okay, this is all new, I don't understand this. You read about it the second time, it makes a little bit more sense. It may take a third or fourth time. So the repetition is going to be helpful. So I would encourage you, again, to use all your resources that you have available to you. All right, so last time we had a little introduction. We talked kind of how we're going to approach immunology, like first line, second line of defense, third line of defense. Let's break into that first line of defense. Remember, this is part of our innate immunity. So this is our non-specific immunity. So generally, every time these mechanisms encounter a pathogen, it's going to be the same defense. Okay, there are some exceptions to that, but for the most part, same. Our body's not changing our response to the invaders. So our first line of defense is mostly physical barriers that the pathogen would encounter when it's trying to gain access to our body. Okay, and if our first line of defense fails, then we go back to our second line of defense, which we'll get to next. Okay, so let's talk about some of those barriers at our portals of entry that we talked about. Skin, we mentioned before, is a really very effective barrier. It's not a great portal of entry for pathogens, and some of the reasons is, let's look at the composition of the skin a little bit more closely. So the outer layers of the skin, the outer epidermis, there's many layers there of cells, and the outermost layers, we have several layers of dead cells. So remember, pathogens have to adhere to cells first before they're going to infect them. If they're dead cells, they have a much harder time doing that. And those dead cells are continuously being sloughed off. Okay, we call that process desquamation. Okay, the continual loss of those layers of skin. Okay, so if pathogens do attach to them, basically those skin cells come off. What happens to all of our skin cells that we're continuously losing? Well, they end up as part of dust, And, where does all that dust go? Well, a lot of it gets eaten by dust mites. Okay, and about a third of us are allergic not to dust or dust mites, but a protein in the feces of dust mites. Okay, so that's why my nose is always running. I'm always up here blowing my nose because I'm allergic to feces. It's a wonderful thought that we're surrounded by that all the time, but that's the world that we live in. Okay, deeper into our layers of the skin. This is a picture that I showed briefly last week. We've got some cells called dendritic cells that we're going to talk more about shortly, so stay tuned. These are phagocytic cells that are patrolling the deeper levels of our skin there for pathogens. We've got sweat glands. Okay, so down here, the sweat, okay, sweat glands secrete sweat. Sweat is fairly salty. So, if you've ever licked your hand after you've worked out or whatever, you could probably guess that. And we know when cells are in salty environments, they lose water, they dehydrate. Okay, let's see. Also in our sweat, there are antimicrobial proteins. That's a fairly general statement. We won't get into any more detail about what exactly that contains. Peptides that act fairly non-specifically against a wide range of pathogens. Also, there's lysozyme in sweat. We've mentioned that chemical a couple times before. That's an enzyme that breaks the bonds between the sugar subunits in peptidoglycan. Okay, so we're going to see lysozyme in several different places today. Also deeper in the skin, we have sebaceous glands. And I think my arrow here didn't quite make it over. I think this is the sebaceous gland over here. Of course, they secrete sebum, which is a chemical that its main job is to keep our skin pliable. Okay, so that's its role. It also lowers the pH of the skin. Right, so all in all, our skin a fairly inhospitable place. It's salty, the pH is low, we've got some other chemicals going on there. Of course, if we get too much sebum production, that's not good. That clogs up our pores and that makes us more likely to have infections like acne. There are some potential for pathogens to infect hair follicles, but for the most part, this is, again, a pretty inhospitable place, unless you get some kind of cut in the skin there that obviously would be a better portal of entry. Okay, we talked about mucous membranes are a little bit easier. We don't have quite as many defenses there, more common portal of entry for pathogens. Remember, mucous membranes are going to line any of our openings to the outside. So urinary tract, digestive tract, reproductive tract. We have a fairly thinner layer of cells than what we do on our skin. And those cells are tightly packed, but they're alive. Okay. Even though they're alive, they are still constantly shedding and being replaced. So that's one mechanism, again, to keep pathogens from attaching. Again, not as happening as much when those cells are dead, but those are still short-lived. We have stem cells that produce a couple of different kinds of cells along our mucous membranes. Goblet cells. These are cells that produce mucus. Okay, so your book didn't provide a picture of this. This was the best one that I could find here. Here's some goblet cells, and you can see the mucus secretion there. Of course, the mucus is designed to trap pathogens and eventually kind of wash them away. And I have read that we produce as much as 1 liter of mucus a day, which is astonishing to think, like, where does that all go? Apparently we swallow a lot of it, but with all our mucous membranes, you know, a lot more than what maybe you would think we do. Okay. And on the epithelial cells lining our respiratory tract, we also have ciliated columnar cells. Okay, so you can see them over here. All right, they've got cilia at the ends of them that are in the respiratory tract. And of course, the movement of the cilia, wave-like movements, helps to dispel those pathogens caught in the mucus. Okay, so remember we talked about flagella and cilia, bacterial flagella, right, move in circles, eukaryotic flagella move in a wave-like motion, just like the cilia, so that makes sense. Okay, if you are a smoker, your cilia don't work as well. Okay, so that's one of the reasons that smokers are more susceptible to respiratory infections. Of course, that's kind of a scary thing right now in the midst of a pandemic of a respiratory infection. And I was trying to find some information about if smokers are more likely to get COVID. There hasn't been any like peer-reviewed articles done as far as I can tell, but the feeling is probably yes, they are more likely to get COVID. Of course, smokers are more likely to have, you know, more lung issues in general that would make people more disposed to have COVID worse. Okay, but that's, again, something to think about. Okay. And in general, we think the smoke limits other aspects of our immunity too, like production of different kinds of cells, productions of cytokines. So, okay. All right, so, and there's some antimicrobial chemicals in that mucus as well. Again, we're not gonna get too specific here. We see lysozyme again. All right, so thinking about other things, kind of barriers to infection. We talked a little bit about our normal microbiota back last chapter. Thinking now, how does our normal microbiota help protect us? Well, we kind of talked about this in general before, but the concept of microbial antagonism. I don't think we used that word before, but that just means our normal microbiota basically outcompete invading pathogens. Okay, so because we already have lots of bacteria, say on our skin, any pathogen that might come and try to adhere there has to compete with our normal bacteria for nutrients, for space. Okay, so that's again a good thing. One way that our bacteria protect us. This is a really interesting area of study right now. That you think about it, why does our immune system tolerate our normal microbiota? It's really kind of a mystery how that works, but we do know that our bacteria that we normally have kind of help stimulate our immune system, not too much, but in a way that kind of keeps our immune system primed. And we know that because we've done these interesting studies with mice that are born into a sterile environment and then raised in a sterile environment. And I don't know how they, you know, oh, well, the mother was sterile. Okay, well, then how was that? You know, I don't know how they were able to get a mouse sterile inside and out. But apparently they've done that. And they've looked at these mice that don't have any normal microbiota. They don't grow normally. They're much smaller, kind of scrawny looking, and they just don't seem as healthy in general. If they are exposed to an organism, to some pathogen, they have a very poor immune response to it and oftentimes will die. So there's something about having some bacteria always present that is necessary for our immune system to really work properly. And we have a lot to learn about that, but there's some really, again, it's a really interesting area. And we've known for a long time that bacteria, especially in our large intestine, helps us digest our food, produces some vitamins for us. B5 is one that's supposed to help in glucose metabolism. So I don't know if that's some kind of a coenzyme for some enzymes in metabolism. Folic acid production by bacteria in our gut. Of course, we need folic acid to make DNA, just like bacteria do. We get a little bit, some of the extra that they make. Some precursors of vitamin K are produced by bacteria in our large intestine that's necessary for blood clotting. So we definitely benefit in lots of ways from our normal microbiota in several different ways. Okay, so just to finish up our first line of defense, these tables I wanted to talk about here, not for you to memorize, but it's just sort of interesting to see how some of our normal body processes have, you know, the main reason that we have that process maybe is not to protect us from pathogens, but it does that in addition to its normal function. So, you know, something like saliva, if we look at some of our bodily fluids, obviously saliva is necessary for us to, you know, wash our food down into the digestive system. There's digestive enzymes in saliva, but it has antimicrobial purposes as well. There's some antimicrobial chemicals in there. Here we see lysosome again, some other enzymes. We know that people that don't produce the normal amount of saliva, it's called Sjogren's syndrome. These people that have this have a lot harder time with tooth decay and gum disease. Okay, so there again are antimicrobial things in saliva that help protect us from tooth decay. And incidentally, you know, when we sleep, we produce a lot less saliva. So that's why it's really important to brush your teeth before you go to sleep. Because again, we don't have that saliva, as much saliva protecting our mouth from those bacteria. Obviously, stomach acid, low pH, a common thing that we see, a recurring theme here that you'll see are strategies to sequester iron. Okay, so we have lots of chemicals in several different fluids that act to sequester iron. Iron is often a limiting nutrient for bacteria when they're trying to infect a host. So we have a lot of signals in our body that kind of signal ourselves to take up any available iron so bacteria don't have access to it. My daughter has been fairly, how do you say this? She runs a lot, okay? And so for many years, she has taken an iron supplement because that's a common thing with female runners to be low in iron. And so she's tried to take a supplement just to kind of head that off. I noticed in her competing days in college that she was getting sick quite a bit. And, you know, strep throat, lots of different things. And I suspected that maybe it was because of that iron, you know, making a lot more iron available to invading pathogens. And I read something about that not long ago, that if you are taking iron, you're a little bit more susceptible to some bacterial infections. Now, if your doctor says take iron, don't stop because I said that, but it's just kind of an interesting thing. Let's see. So again, don't memorize these, but again, here you see in lots of our secretions strategies for, you know, iron binding and sequestering iron. Okay, binding iron. All right, so that was pretty brief sum up of our first line of defense. Okay, hopefully doing fine so far. Okay, so again, once our first line of defense is breached, then we call into some strategies of our second line of defense. And remember, this is still our innate immunity Okay, second, third, fourth time we encounter pathogens, these processes act exactly the same, usually. Okay, so we're gonna take a look, again, we are in chapter 15. We're gonna look pretty closely at some of the cells that function here, lots of white blood cells to talk about. We're gonna be looking at some antimicrobial chemicals, like complement proteins, how they work, just kind of a real brief overview of cytokines, and we'll talk more about the processes of inflammation and fever and how those help protect us. Okay, so this is the figure in your book where figure 15.4. All right, so this is labeled in your book, but I think it's helpful for us to kind of label these ourselves whenever possible, do something active, right? So these are some of the cells that are involved in our second line of defense. So we're going to start out with some stem cell in the bone marrow. Okay, and by a process of differentiation called hematopoiesis. So again, this is just kind of means differentiation. Okay, cells are beginning to specialize into three types of some stem cells: erythroid, myeloid, and lymphoid. And this is still in the bone marrow. Okay, again, by specialization, hematopoiesis, those cells or precursor cells become all these different cells that are in our blood. So erythroid stem cells become red blood cells. Of course, their function is to carry oxygen. As far as I know, they don't have any immune function. Okay, so we're not going to talk about them anymore. Lots of different cells descended from myeloid stem cells. We've got platelets, and their main function is to clot the blood. Apparently, we think now that they do play a role in some immune function, but it's probably slight, and we're not going to really bring those in and talk about them anymore. And then we have three different kinds of, these are white blood cells with granules in them: basophils, neutrophils, and eosinophils. And you don't have to be able to recognize which of those is which. Okay, these are all considered granulocytes because they've got these little granules with digestive enzymes in them. So we're going to talk about them each specifically in a minute. And then you have also descended from the myeloid stem cells, monocytes. The monocytes circulate in the blood. When they move out into the tissues, they differentiate into two kinds of cells: macrophages, which you probably heard of, and dendritic cells, which we mentioned briefly as patrolling the skin. Okay, and then lymphoid cells, we got three kinds of lymphocytes. Sorry, good question. A monocyte is an agranulocyte. Okay, so let's see. I was trying to figure out how I can label that. Sorry, granulocytes. This means they don't have granules. So monocytes, macrophages, dendritic cells, agranulocytes. Sorry about that. No granules. Okay. Lymphocytes. This is our B cells and our T cells that are playing a big role in adaptive immunity, as well as natural killer cells. We often call those NK cells. They function more in innate immunity, and we're not going to talk about them very much, but they are considered lymphocytes as well. Okay, so all of our lymphocytes, the monocytes, macrophages, and dendritic cells do not have granules. They are agranulocytes. Okay, and that looks a little sloppy. The granulocytes and the agranulocytes, those are all called leukocytes. Okay, so grans and agrans, leukocytes. Okay, we've got lots of names for different things, also called white blood cells. So definitely the potential to be confusing there. Let's talk about some of these cells in a little bit more detail. Yep, wait just a second. I know if you're trying to type that out, that might not be easy. But again, that is labeled in your book. So I don't think your book is very clear about where macrophages and dendritic cells come from. Okay, so again, you don't need to, I wouldn't show you a picture and say, you know, what kind of cell is this? I'm certainly not that good. Take a good histology class if you want to be good at that. Let's talk first about neutrophils. These are the granulocytes that we're going to spend the most time on. Neutrophils also have a second name. They are also called PMNs. Oops. Okay, and that stands for polymorphonuclear leukocyte. Okay, so that's where the PMN comes from. Okay, so you can see the nucleus here has many lobes. So that's where the name comes from, polymorphonuclear leukocyte. Now, actually, basophils and eosinophils have many lobed nuclei as well. But because we have so many more neutrophils than basophils or eosinophils, the term PMN is usually just referred to neutrophils. Again, sometimes you could see some argument about that, but again, because there's so many more neutrophils, we usually use that term synonymous with PMN. I always call them neutrophils though, but you will see that term PMN other places, PMN. Okay, so about 50 to 60% of the white blood cells in circulation are neutrophils, so they're pretty popular. One of their primary functions is phagocytosis. So they are phagocytic, and they're highly motile. Okay, so these are moving around in the bloodstream. When they get the correct signals to do so, they're leaving the bloodstream and going out into the tissues. They move by amoeboid motion, and they are considered kind of, you could call them the first responders to the site of an infection. Okay, so this is the emergency squad. Okay, they chemotax to the site of an infection. They've got the correct chemical signals that they're following. And these cells, there's so many of them, they're usually the first one there. But they are short-lived. Okay, they usually only last a matter of hours when they're out in the tissues, actually acting on pathogens. We'll talk about why that's not such a bad thing. Okay, because they can have effects that are damaging to our cells as well. So that's really not such a bad thing. Okay, how do they act? They actually act in 3 different ways. Like I said, their primary function is phagocytosis, and we're going to look at the steps of phagocytosis in a little bit more detail soon. But there's a link here to a YouTube video that's just a couple minutes long, and it shows some neutrophils going through the bloodstream and how they might, it's a, an animation, a drawing, how they might move into the bloodstream. It's kind of helpful to watch. Neutrophils also act on pathogens by degranulation, right? So these granules with digestive enzymes in the cells are released onto a pathogen, and what's in those granules, things that would harm the pathogen, also inflammatory mediators. So we're going to talk more about inflammation and the good that inflammation actually does. So stay tuned for that. Okay, but that's why it's best if they're short-lived, because when they degranulate, it harms our cells as well. Too much, if they continuously were degranulating, too much inflammation is bad for us. So we want to kind of dial in that response. They live for a couple hours. If we need some more, like, we recruit more and make more. A third way that neutrophils act on pathogens has been fairly recently described, the formation of nets. So that stands for neutrophil extracellular trap. Okay, so basically neutrophils can act kind of as kamikaze warriors. They basically can lyse their DNA. And if you've ever done the mini prep in the lab or ever isolated DNA from anything, it's kind of a sticky substance. Okay, so they can kind of blow up their DNA and throw it out on some microbes. So in this picture shows this is a Klebsiella pneumoniae. That's an organism that we use in lab that's infecting the lung tissue of a mouse. Okay, where's the neutrophil in this picture? It's basically blown itself up and made this kind of meshwork in order to immobilize that pathogen. Okay, so what good is that? Well, now other cells can come in now that cell's not moving around. And there's also some antimicrobial compounds on the nets that's going to help kill that pathogen. Okay, here is kind of an interesting picture of, this is supposed to be a neutrophil phagocytizing some anthrax bacteria. It kind of reminds me of a python trying to eat an antelope or something like that. Typically, a neutrophil you would think would be way bigger than the bacteria. So that kind of surprises me, but it's still sort of an interesting electron, scanning electron picture there to look at. All right, eosinophils and basophils, I don't have as much to say about them. They are, again, much less common than the neutrophils in the bloodstream. Primarily, they both act by secreting the granules, although they may be phagocytic. Rarely, we think that that's not normally the way that they function. Okay, we think of the eosinophils primarily acting on eukaryotic pathogens, okay, especially fungi and helminths. So helminths are multicellular worms, like flatworms, like tapeworms, fluke worms. And this is a picture that I don't have this cited because actually where I found this picture was on Pinterest, of all places. And guys, if you don't know what Pinterest is, ask your mom, maybe she'll know. So this picture is too good not to show you, though. Here's some eosinophils that are surrounding a fluke worm. Those eosinophils are releasing their granules. It takes a lot of release, a lot of cells to actually probably have any effect on such a big organism. So they're degranulating, again, digestive enzymes as well as inflammatory mediators, which is going to act to call other cells to the site here to act against that big pathogen. We also know that if we have too many eosinophils, then that can be a sign of some kind of allergy, some kind of inappropriate immune response. There's a lot you can tell by doing a blood test and counting the proportions of certain kinds of cells in your blood. A lot of hints that gives us as to what's going on in the body, if it's an infection, maybe what kind of infection, or if it's not an infectious agent at all. Okay, so we don't know as much about basophils. Again, we think that they have kind of a similar role as eosinophils, but maybe not as much. Okay, it's just, we don't know as much, but again, they're a granulocyte. They've got these, lobed nuclei. Okay. Questions so far? Okay. All right, moving into the agranulocytes without granules. There's lots of some monocytes circulating in the blood. And like I mentioned before, when those move into the tissues, they start to differentiate. Macrophages, I often use the shorthand like that, macrophage. So there's a couple times where I've written that and maybe not explained that, but that's kind of a shorthand for a macrophage. Their primary function is phagocytosis. Okay, and like I mentioned, we're going to talk more about the stages of that. They also are important to process and present antigen. And that's a very important process. So basically, here's our macrophage in getting ready to engulf some, bacteria cell or whatever. So here's that bacteria. It's within a membrane there. The lysosome fuses with that, and basically that bacteria gets all kind of chewed up. Well, that macrophage's job is not done. It takes part of that chewed up antigen and displays it on its surface. Okay, we're going to talk a little bit more about the mechanism of how that does that. But that's very important, because if the macrophages are not going to be enough to take care of that infection, this is how it calls into help our third line of defense, our B cells and our T cells. Okay, so it goes, presents antigen in the lymph nodes, maybe somewhere, to get help from B and T cells. Okay, let's see, they communicate with cytokine signals. Again, we're not going to be too specific about what those signals are, but that's happening all the time. They are also considered the garbage collectors of our body. So basically, they're patrolling around and eating up dead cells or dying cells. Okay, whoops, garbage collectors. That's what I meant to say. Okay, and they're pretty long-lived. Unlike neutrophils. I talked about how the neutrophils degranulate. That can be bad for our cells as well. So macrophages, they're out just patrolling. It's good that they're long-lived because they're not doing any harm to us generally. Okay. Monocytes also can differentiate into dendritic cells. I don't think your book shows any picture. That root there, dendrite, I think that's Latin for tree, maybe. You might think of a nerve cell as being a dendritic cell. So that's where the name comes from. These cells are also phagocytic. They also process and present antigen. Okay, we're going to further define that term antigen, but that's basically just a piece of a pathogen that our immune system would recognize as being foreign. Okay, so we're going to talk more about that. But dendritic cells are really good at presenting that antigen because they have these kind of long arms to do that. So again, here's a little short YouTube clip that kind of compares macrophages and dendritic cells. This is a little cartoon, and it talks about this process of presenting antigen in kind of a basic way, but it's kind of a fun little animation. All right, and lastly, I'll just briefly talk about the lymphocytes. We're going to get much more detail about their function in the coming weeks. Lymphocytes are mostly in the lymph. Okay, they are some also circulating in the blood, but again, mostly in the lymph nodes. Okay, so B cells, they differentiate plasma cells, and the main function of a plasma cell is to produce antibodies. Okay, so we talk about B cells being important in humoral immunity. Okay, so that's the kind of adaptive immunity. Humoral means in bodily fluids. So antibodies in our bloodstream, there are other places as well, but that's what that term humoral means. Okay, and T cells, there's a couple different kinds of T cells, cytotoxic T cells and helper T cells. Cytotoxic T cells act to kill infected cells. We'll go into a little more detail about how they bring that about, especially cells that are infected with some intracellular pathogen, like a virus. Okay, that's our cytotoxic T cells are mostly acting on virally infected cells. We'll talk more about that. T helper cells coordinate many aspects of immunity. They're kind of like the general in charge, basically. So they're acting on a lot of different cells. And all in all, we would consider this cell-mediated immunity. Okay, that's mostly carried out by T cells. All right, and then I just mentioned briefly before these natural killer cells. Kind of an interesting name. We called them that because at first we didn't understand these cells they act on virally infected cells, and we didn't understand to begin with how the cell knew that they were infected with a virus. It's inside the cell. So we called them natural killer cells just because, well, somehow they know that they're supposed to kill these. We understand that mechanism a little bit better now, but the name stuck. These are considered part of our innate immunity, where B cell and T cell action is, we would consider that adaptive immunity. And again, we're not really going to visit NK cells too much, but we think they're important mostly for virally infected cells. All right, so we have talked about phagocytosis a little bit when we were talking about anti-phagocytic factors last week, but I want to go a little bit more carefully through the steps of phagocytosis. Remember, our phagocytic cells, macrophages, dendritic cells, and neutrophils are going to be our main, sometimes they're referred to as professional phagocytes. Okay, so those are the kinds of cells that are mostly performing phagocytosis. If we looked at the stages of phagocytosis, if we break that down a little bit more carefully, first thing is chemotaxis. So a macrophage, for example, has to move to the sites of where it's necessary. It has to follow chemical signals and get to the right place where it's supposed to be. Sometimes we kind of forget that. They're not just always right there where they're supposed to be. Okay, and this recognition, whoops, I don't want to do that. Okay. It's important, again, to think about, we don't want our macrophages engulfing, our cells. Okay, we want them to recognize cells that are pathogens and only act on pathogens, right? We don't want them acting on our own cells. So they have to have a mechanism to understand, yes, this cell is worthy of my engulfment. Okay, so call that recognition, and they're going to have these receptors called toll-like receptors on their surface. It's not an intuitive name. It's a German word, toll, and it's named because the researchers realized, hey, this is like a receptor that we see in fruit flies. It must be like those, so the toll-like receptors, okay? But we abbreviate them TLRs. Lots of abbreviations. To read a research paper that has anything to do with immunology, you kind of have to be fluent in those abbreviations. So TLRs, toll-like receptors. Okay, these are transmembrane proteins of phagocytic cells. Okay, we've got a picture that I'll show you in just a second. These receptors recognize broad-based patterns. We call those patterns PAMPs. So that stands for pathogen-associated molecular patterns. Kind of a weird abbreviation, PAMPs. Okay, so these are broad-based patterns that are typically seen on pathogens, but not in our body. Okay, so they're not terribly specific. They're going to recognize things like lipopolysaccharide. Okay, only gram-negatives have that in their cell wall. Okay, we don't find that in our cells. Maybe something like teichoic acid or lipoteichoic acid that would only be found in gram-positive cell walls. Maybe some peptidoglycan, right? Only bacterial. So these are, again, broad-based patterns that you would see in lots of different pathogens. And when these receptors recognize these patterns, that not only tells the phagocyte, yes, this is something that should be engulfed, but it kind of trips an alarm for lots of other aspects of immunity as well. So send out chemical signals so that other cells would come and help take care of the pathogen, for example. It might call complement proteins into action, more inflammation, and I know we haven't talked about that yet. Okay, so here is, your book doesn't provide a graphic to visualize this, so this was the best one that I could find. There is about 10 different toll-like receptors. That number seems to be kind of edging up, like they discover a new one every once in a while. There might be a couple more than that now. But the people that, for instance, discovered toll-like receptor 4 that recognize gram-negative bacteria, they got a Nobel Prize for this. So this is important work. And you don't need to memorize, oh, TLR5 recognizes flagellin. Like, that's not the point. You don't need to memorize switch does switch. I don't have that memorized. But again, recognizing broad-based patterns that we often see in pathogens. So these receptors would, send signals to the cell, yes, engulf this pathogen. And then actually there's some toll-like receptors that get expressed on the phagosome, on that membrane, once it's engulfed. Okay, and these are going to recognize things like double-stranded RNA. That's something that we never find in our body, or single-stranded DNA. There's not one for that. But there are subtle differences in, say, bacterial or certainly viral nucleic acid that our toll-like receptors can recognize, like different methylation patterns, for instance. Okay, and then of course, these when these receptors are tripped, that sends chemical signals again, for other cells to come and help, or other aspects of immunity. Okay, so that toll-like receptors, if you look at this figure from your book here, the events in phagocytosis, we've got a little inset here where they've got looks like a transmission electron micrograph showing like this is the way it would really look, but that's really not clear, so we're going to draw the rest of the figure. They talk about chemotaxis of the microbe here, and adherence / recognition. Talked a little bit more about recognition. Okay, and that ingestion, sometimes there are factors that can make our phagocytic cell more likely to be able to ingest the pathogen, because as we saw before, there are lots of virulence factors that bacteria have that would make them more likely to avoid phagocytosis. Okay, we call them anti-phagocytic factors. All right, so we have ways then, it's kind of like an arms race, we have ways to try to still be successful in phagocytosis. So any factor that's going to enhance phagocytosis is called an opsonin. Okay, it's kind of a weird term that is easy to trip over. But an opsonin is something, again, that increases the chances that phagocytosis will take place. So some examples of opsonins, antibodies can bind to the pathogen and they make it more likely for the pseudopods to attach to it, and as well as complement. So make a note of that. We will talk about those functions of complement in antibodies when we get to them. All right, so again, lots of interwoven things. We have to start somewhere. So we'll talk more about opsonins. Okay, so once the, say, neutrophil or whatever is successful in engulfing that pathogen, here's in the phagosome, right? That phagosome fuses with the lysosome, right? And the lysosome releases its antimicrobial chemicals, and we call that the phagolysosome. All right, so what sort of chemicals are in the lysosome that make it really effective at killing whatever that pathogen is? Here's some examples. There are at least, I think I wrote this number, I think there's like 40 different enzymes that we might see in a lysosome. So this is just an example of some of the things. Lots of different digestive enzymes. All right, so proteases that digest proteins, lipases that would digest lipids. You see that lysozyme again that acts on bacterial peptidoglycan. Nucleases digesting nucleic acid. Okay, those are just some examples. There's some other really cool things that can go on in there. There's respiratory burst enzymes. We've mentioned a lot these forms of toxic oxygen and how dangerous they can be to a cell because they're pulling off electrons from other molecules. Okay, so they're oxidizing agents. We can actually make toxic forms of oxygen in our phagocytic cells that would, again, act to kill invaders. And in neutrophils, they actually have this enzyme called myeloperoxidase that actually they're able to produce hypochlorite. That's bleach, right? So we actually can bleach invaders in there. And this is only a very small amount. You would think, you know, that might get released inside the neutrophil, but apparently it just acts really locally and doesn't have any effects on the cell, and even, you know, once it's released into outside of the cell. Okay, this has a heme protein in it, and so pus, when you think, well, what is pus? It's lots of kind of dead cells and fluid. Sometimes pus looks a little greenish. That's from this heme protein of myeloperoxidase. So just kind of interesting. All right, so cutting back to this, most of the time, the pathogen is killed and then it's, released to the outside. Now, bits of that pathogen, like I spoke of before, if you're talking about a macrophage or a dendritic cell, bits of that pathogen are going to be displayed on the surface of the macrophage. Okay, but this drawing doesn't deal with that, and we'll talk more about that later. Okay. One more thing to mention is that phagocytes, especially macrophages, can be activated. If they're encountering a particularly robust pathogen, then they might need to kind of step up their game a little bit. Okay, so especially macrophages, this happens with, and they get activated by cytokine signals. So what happens when they get activated? They get bigger, they move faster, they make more lysosomal proteins, increased metabolism, they need more energy, they're sending out more cytokines, so they're just in general more effective. The analogy that works for me, being a child of the 80s, is Pac-Man. Okay, so if you think about Pac-Man as being a macrophage, it's normally going about its business, eating the dots, cleaning up dead cells from all around our body. We have lots of macrophages in our spleen, in our lungs. But if the macrophage gets activated, if it eats one of the big yellow dots, it gets bigger, it moves faster. Now Pac-Man can eat the ghosts even. Okay, so it's just sort of a new level of energy. The problem is, breaking away from that analogy there, even though it is more effective in killing invaders, it comes at some cost because it's going to give an increased level of inflammation. We get too much inflammation, that's bad for our cells as well. So it does come at some cost. We don't maybe want this happening all the time. Okay. And I didn't post this because this video, I was so sad. This video got taken down from YouTube. I don't know why it did, but I had this great 1 minute animation of these activated macrophages moving in time lapse and engulfing these little yeast cells. It was really kind of the coolest thing, but unfortunately I didn't post it because it's been taken down. So I don't know why I felt compelled to share that with you, but there you have it. Okay, complement. So I assume that probably some of these aspects of immunity you've had, maybe a little bit of anatomy, talked about cells. Complement seems to be a topic that's going to be new for most of you. There's only a couple pages in the book that talk about complement. I would just certainly encourage you to do that. I always have students ask me, what do I need to know about this? And I've tried to be especially clear in my learning objectives, what I really want you to know. So by all means, ask me that, but look at the learning objectives first and see if that answers your question before you ask me. What is complement? It is a series of about 20 proteins that are always present in the blood. They're actually made in the liver, but they circulate in the blood. So these are proteins that complement other aspects of our immunity. Okay, so they can act directly on invaders, even to kill invaders, but they have a major role in, again, helping other aspects of our immunity. Okay, it's considered, again, we're still at our second line of defense, our innate immunity, non-specific immune reaction, and it's considered a cascade reaction. So that means one protein will cause a change to another protein, which causes a change to another protein. Okay, it's like the rock that starts out rolling at the top of the hill and becomes, an avalanche by the time it's the bottom. Okay, so one thing causes another, causes another. Okay, functions of complement. Some of these complement proteins act as opsonins. Okay, they act to increase the chances of phagocytosis. Some complement proteins act to increase inflammation. And chemotaxis. We'll understand in a little bit how those two things really go hand in hand. The inflammation increases kind of the amount of fluids in the tissues, and that can help signals get to where they're supposed to go. They can also, complement proteins can also form membrane attack complexes. And we abbreviate those MACs, membrane attack complexes. Okay, so some of these proteins can come together and actually poke holes in membranes. Okay, they would poke holes in our membranes if our cells didn't have proteins on our surface to downregulate complement. Okay, so I'll add here, especially it's going to work on gram-negative bacteria. Okay, so gram-positives obviously have the thick layer of peptidoglycan. These proteins don't penetrate very well through that, but for gram-negatives can go through the outer membrane and the inner membrane. Sorry about that noise outside the door there. Thank you for shutting the door. Oh, a big sign outside there. Please don't play basketball. Please don't do this. Please don't play music. Wouldn't be that hard. Okay. Thank you. All right, so a little overview of the complement cascade. Again, this is not from your book, but I just wanted something a little bit basic to start out with. Here's a bacterial cell, so some pathogen. How the complement cascade gets activated, there's a few different ways that can happen. I'm going to skip over that for now and come back to that. But in this case, there's something on the bacterial cell that's acting as an antigen. Remember, an antigen elicits an immune response. And an antibody, in this case, has bound to it. And then the first complement protein recognizes that, and it becomes activated. So when it becomes activated, now it's acting like an enzyme. And that complement protein acts on others to split them, which acts on others, and kind of so on and so forth. These little bits that are kind of coming off here, nothing is wasted. Some of those act as opsonins or to increase inflammation. But lots of the bits that we kind of end up with come together to form these membrane attack complexes. Okay, and again, you poke holes, get enough holes poked in a bacterial cell, it's not going to be able to make energy. Things are going to be, coming out of the cell that shouldn't be. We got water rushing in, turgor pressure, and the cell is likely to burst. So you get enough of those, and then it's going to be really bad for the cell. Okay. All right, so I kind of went back and forth about whether to draw this out myself or to use the book's drawing. I think in the interest of time, I'm just going to talk you through the book's drawing here. It's for the most part pretty good. And I'll try to point out, again, the spots that I think are especially important, because it's not every detail you need to know here. But so here's the first complement protein, C1. Okay, so it's activated here by an antibody bound to an antigen. All right, again, we'll talk more about that. This is called the classical pathway, when you have activation by an antibody antigen compound. Okay, so that C1 becomes activated what does it do? It acts as an enzyme on C2 and C4 and splits those proteins. When you have a split protein, they name the fragments A and B. So C2 splits into C2A and C2B, C4A and C4B. Some of those fragments come together to form another enzyme. And I get mixed up if it's the B or the A of which one or the other, that's all right. But this is important enough that we give these two fragments that come together their own name. We call this C3 convertase. Okay, and C3 convertase acts on complement fragment C3 to split it into C3A and C3B. Okay, anything with a fragment A, you could think allergy. Okay, so that's going to increase inflammation and chemotaxis of cells to the area. Okay, so that's what the C3A does there. The C3B, okay, B, you're going to think bind. Okay, so the B fragments act as opsonins. Okay, so this C3B, some of that C3B will come up here and bind to the pathogen. Okay, so when it binds to the outside of the pathogen, then you can understand there are receptors for that in our phagocytes. Okay, now they can grab a hold of that, okay, and ingest it. It's like if you're a rock climber, you don't want to go up a smooth wall. You want something to hold on to. Okay, so that's acting as an opsonin. But some of that C3B binds back to this, original C3 convertase. And now it acts on another complement protein, C5, and splits C5. Again, C5A triggers inflammation. The C5B, B binds. It's shown right up here. It's binding back to that pathogen along with lots of other complement proteins, C6, C7, C8, and lots of C9s, okay, and making that membrane attack complex. Okay, so the parts that I want you to remember, again, the A fragments, more inflammation, the B fragments, opsonins, they're binding. Okay, so if we, so here's a transmission electron micrograph of a cell that's got problems because it has lots of holes poked in it by those complement proteins, those membrane attack complexes. And again, this is most effective on gram-negative bacterial pathogens. Just can't poke holes in that peptidoglycan of the gram-positives. Okay, so if a person gets certain kinds of gram-negative infections fairly often, then we start to think that maybe that person has some kind of complement deficiency. Meningitis is kind of a big one. It's a fairly rare infection caused by a gram-negative organism. And if a patient, to get it once is pretty rare, but then if somebody gets it again in their lifetime, then again, they start looking to see if they've got some kind of complement deficiency. Okay, so let's finish up with how complement becomes activated. There's 3 different ways that those proteins start to act as enzymes. The classical pathway, this involves antibody. Okay, so that's typically, if this is a new pathogen that the host hasn't been exposed to, the first complement is not going to get activated by this pathway because it takes some time for the cell to make antibodies to a new pathogen. It might take a week or more. So this is kind of a crossover. We think about complement as being innate immunity, but antibodies definitely specific adaptive. So there's a little crossover there. A patient could have some circulating antibodies, maybe from an old infection, and then potentially it could activate it more quickly. The alternative pathway, we'll say it's activated by pathogens. It's a little bit more complicated than that. I'll show you a picture in a minute. And the lectin pathway, this is activated by polysaccharides that we only see on microbes. So microbial polysaccharides, sugars. Sorry, my handwriting's not so good there. Microbial polysaccharides. Okay, and here's a little link here that I just stuck on this page. Again, I'd love to show these in class. It's this site called Help Hippo, and it kind of runs through this complement cascade kind of quickly, but it's kind of again, it's always good to see these things visualized. Okay, so this is your book's graphic on how the 3 different ways that complement can be activated. What I want to emphasize here that no matter how complement is activated, we get the same results. Okay, so let me write that. So no matter how it's activated, the outcome is the same. Okay, so we get the same benefits from complement, the same membrane attack complex. It doesn't matter which of the ways it was activated. Okay, so classically, again, with some kind of antibody, it's drawn here with the IgM, which is usually the first antibody produced in an infection. We'll talk about the kinds of antibodies soon. With this alternative pathway, basically that C3 complement protein is actually always being cleaved kind of at a low level. And that happens spontaneously if that C3b happens to be in close contact with some kind of microbe. It will bind to it on its own. And if there's some other factors present that we're not going to go into detail about, then it will act as C3 convertase and start the complement cascade. So again, that's kind of happening at a low level, and if there's the right things around, then it will become activated. The lectin pathway, I think this is the most common way that complement is activated. So here's some sugars like mannose on the surface. That's very common for mannose to be on the surface of bacterial cells. And lectins are proteins that we make in the liver that bind to mannose. Okay, so when lectin is bound to mannose, then that acts again as a convertase to cleave C3 and start the rest of the cascade. Okay, I think that's all I had on complement. Do we have any questions? Yes, so say it real loud. Yeah.
Is that one specific pathway then?
Okay, so the question is this a specific pathway? No. So no matter if it's, you know, we would get all of this the same, even if it's by the alternative pathway or the lectin mannose pathway. Does that answer your question? So yeah, this part right here would be a little different. There wouldn't be antibodies here. Does that answer your question?
Right. So all of it would not be included in the pathway.
Right. So we wouldn't necessarily have the lectin and the mannose and the antibody every time. We'd have any one of those 3 ways would be enough to start the complement cascade. Yes, yes, but they all do the same thing. Yep, yep. Okay, have a good weekend. I will see you on Tuesday.